Hi everyone, my name is Claire Oswald. I'm an associate professor in the Department of Geography and Environmental Studies here at Toronto Metropolitan University. I am the co-principal investigator on a project called Wastewater Surveillance for COVID-19 in the city of Toronto, and I collaborate with Dr. Kimberly Gilbride and other researchers in the Urban Water Research Centre on this project. The goal of the project is to track the prevalence of COVID-19 in the city of Toronto through wastewater sampling. We actually collect samples of wastewater from the wastewater treatment plant and upstream community and facility sites, and we extract genetic material of the COVID-19 virus from the wastewater, and we are able to quantify the levels of virus in the areas we're sampling using this method. This information is really useful for public health decision making. It's a complementary data set that can be used to figure out what the trends in COVID-19 are in the city. We're partnering with Toronto Public Health, the Ontario Ministry of Environment, Conservation and Parks, and we're also collaborating with a number of homeless shelters and long-term care homes on this project. Some of the interesting results that we found so far is that the wastewater viral signals match clinical case indicators very well. And we've even been successful in capturing several facility outbreaks before they were known to public health. And certain interventions were put in place to try to mitigate the outbreaks because of our data. Moving forward, we are continuing to provide wastewater surveillance to the province of Ontario in a service capacity for the next year. And we're also actively conducting research on wastewater surveillance development. So looking at the potential to use this approach for other pathogens such as influenza, and also just looking to understand how different environmental conditions could impact the quality of the wastewater surveillance results. Thanks very much.
My name is Chris Randall. I'm a master's student within the Environmental Applied Science and Management program, and I'm completing my thesis by completing a road salt mass balance or a chloride mass balance within the Black Creek area. So I'm looking at 2 study sites along the Black Creek River here in Toronto, Ontario. The purpose of studying road salt is to identify whether or not the inputs of the road salt being applied to roadways, driveways, and sidewalks matches the outputs of chloride concentrations within the outfalls in my 2 study areas. The importance of this is to identify whether or not there's a hidden storage deposit within these 2 areas that is currently not being identified. By doing this, I am taking the input values and the output values and identifying the winter period, whether or not all the road salt that gets applied over an entire salting season matches the total chloride concentration output during that winter season. I'm hoping to find a balance. However, my prediction is that unfortunately it will be unbalanced. There will be a higher input than there is an output, meaning there will be a hidden storage. I'm working under the City of Toronto, and I am just finishing up my research. My preliminary findings are that there is a lot of road salt entering these waterways, and there hopefully is a little bit of room for reduction, which will eventually reduce the impacts of road salt within our natural waterways. Regarding next steps, we are going to continue monitoring the chloride levels at these 2 sites so we can get a long-term viewpoint on whether or not the chloride trends are increasing or decreasing within these 2 areas. And with that, we will hopefully find that as we reduce our road salt in the future, our chloride concentrations entering our waterways will also decrease.
Hi, my name is Corbin Sparks. I study Environmental Applied Science and Management at the master's level at Toronto Metropolitan University. I am currently working with Dr. Stephanie Mellis on a project involving green walls. So our current project is assessing the airborne volatile organic compound removal efficiency of green walls, along with characterizing their microbiome. So the problem at hand is that in indoor spaces, there are many volatile organic compounds that are in the air due to things like adhesives or fabric treatments. With the green walls, we're able to remove those chemicals using plants and their microbiome. So, in order to actually remove these chemicals from the air using the green wall, we have several things built into the equipment. So there is a watering unit that trickles down water throughout the system into a water reservoir where fertilizer is added to the water in order to keep the plants happy and healthy. In the back as well, there is a fan which draws air into the green wall and through the plant roots, which greatly increases the amount of air that the microbes can interact with, which increases the volatile organic compound removal rate. So we're working with a Canadian-based company called New Earth Solutions. They're the ones who produced the Respira green wall unit that we're using in our tests. And in terms of our future steps with these kinds of projects, we're currently testing what microbes are found within the green wall. And in the future, we'd like to see if we can select specifically for these microbes that do the best at removing volatile organic compounds to increase the efficiency of the green wall's filtration. I'd like to thank Dr. Stephanie Mellis for all of her help throughout this project, along with Dr. Hausner and Dr. Gilbride for providing lab space and technical support. Thank you also to MyTax and New Earth Solutions for providing the green wall to test.
Good day, I'm Barry Orr, and today we're at the Flushability Lab in our Toronto Metropolitan University. And I'm going to show you the research that I'm working on around flushable wipes. Are they really that flushable? So first I have a toilet paper. Now toilet paper, we've always considered it pretty flushable. Well, let's test it out and see what happens. We scrunch it up, drop it in, and we flush it down the swirling bowl of magic. All right, so now we've flushed the toilet, we've simulated our in-house plumbing system and our sewer line. And so this is at the end of the sewer line. And so we've captured the toilet paper, and you can see it started to break apart. So now we're going to take it to another test, which is the slosh box, which will simulate what's going on in the sewer main. So here's our slosh box. We monitor the temperature, we're monitoring the rotations, and we monitor the time. And so we just drop it in there. And as you can already see, we just dropped it in. The toilet paper was falling apart instantaneously. So this is good. Now we're going to leave it and come back to it. We're going to try the disinfectant wipe now. And we're going to put it into the toilet. We're going to flush it down that swirling bowl of magic. It magically disappears, and we'll go catch it on the other end. Now, here's that disinfectant wipe. We flushed it through. We'll put it in, and you can see already the toilet paper has fallen apart. Well, let's just see what happens to that wipe. So now we've tested out a new technology flushable wipe, and we've flushed it through the toilet, flushed it through the sewer line, and we picked it up, and you can see that it's already fallen apart into small little fibers. So let's take it over to the box and compare it. You can see it's already fallen apart so much I can't even get it out. So this is brand new technology for a flushable wipe that is falling apart in the private drain connection. So that's really exciting for our research, because now we know in North America we have truly flushable wipes. There's been a huge debate about what is an acceptable criteria for flushable. Well, now we know that things will fall apart instantaneously, so we're excited. So let's see what's happened here. We've been testing that toilet paper for now a couple minutes. It's fallen apart, no problem there. You see, the disinfectant wipe is not falling apart at all. It's very strong. So that's not flushable. Look at our new technology flushable wipe. It's all into little fibers, like every single little fiber, individual fiber, is now present in that box. So that's exciting. Okay, so a continuation of our research now is going to simulate an actual 8-inch sewer line in the flow. And then we want to see if there's imperfections in our sewers when the products, such as wipes, snag on these imperfections. Do they disperse after a period of time, or actually are they staying within the flow and causing a blockage, or when you mix fat, oils, and grease coming down the drains, a fatberg? So these are things that we don't want to see in the sewer system. So let's see what this simulation does. Okay, we're going to drop the wipe into the sewer line and watch it snag. So you see it snag on that imperfection. And what we're simulating here is that there's a gasket that's offset in this 8-inch sewer pipe, which is a real world scenario for us because the gaskets get offset. We're going to leave it in there for 30 minutes to see if it starts to break apart. Okay, so we took it off the snag for 30 minutes, and as you can see, it hasn't fallen apart. But as part of our research, we're going to take this, we're going to dry it in the oven and weigh it to see if there has been any dispersibility from A, our package wipe, to B, our tested wipe. Okay, so now we're going to try with that brand new technology flushable wipe. We're going to bypass all the testing. We're going to take it directly from the package just to try it and see what happens. Well, as you can see, it already had started to disperse and it fell all apart, didn't even snag. And so that's why we put the sieve there, because it literally fell all apart into pieces, no snag. That's exciting. So we really want to avoid those blockages in the sewer. And as you see, I'm holding one of those blockages from a pumping station in the City of London. I want to thank the Canadian Water and Wastewater Association, the Municipal Enforcement Sewer Use Group, the City of London, and many, many more partners all across the globe on dealing with this research and coming up with a solve on fatbergs and blockages. And this research, I think, is showing that the technology exists to have dispersible products, but it also shows us that products are failing and should never have been labeled flushable.
Hello everyone, I'm Dr. Rania Hamza. I'm an assistant professor at Toronto Metropolitan University, formerly Ryerson University at the Department of Civil Engineering. And my research work is focused on wastewater treatment, wastewater intensification, and removal of conventional and emerging contaminants from wastewater. The research topics that I'm currently working on include a list of contaminants that we call them, or a group of contaminants that we call them together contaminants of emerging concern, or CEC. These are not only one group of contaminants, they are groups and groups of contaminants, thousands of pollutants that actually come up from our different sources, including industrial, domestic, municipal, and they include pharmaceuticals, personal care products, microplastics, the new groups of, or not, I would say the legacy contaminants like the PFAS or per and polyfluoroalkyl substances that are found in firefighting foams, the Gore-Tex or the water-resistant products that we have. So all these contaminants enter the wastewater treatment plant, and these treatment systems were never designed to remove these contaminants. And that's why we do varying, I would say, a degree of removal, and we end up with these contaminants in the effluents or the treated water that will end up back to our receiving water bodies, which are also our source of drinking water. So my work is focusing on how can we remove these contaminants from wastewater, what is the fate of this, and as you can see in my background, our lab, Water Research and Recovery Lab that we have here at the TMU, and we're running different biological systems from suspended and biofilm cultures or microbial aggregates, which we call them, like the aerobic granular sludge, to see the fate and the removal of these contaminants. How can we design a system that will be able to target both conventional contaminants like the organics and nutrients, and also tackle these new contaminants and see how will actually these contaminants affect the performance of the system, and how we can also design systems and optimize systems to be able to better remove them and protect the surface waters. As for the project that we're currently working on, we have 3 main projects that focus on emerging contaminants. One of them is focused on PFAS, which, like I mentioned, the per and chlorofluoroalkyl substances. And Zanina is working on that. She's currently a PhD student focusing on that work, and we're doing that in collaboration with chemistry and biology. Dr. Roxana Suring is co-supervising that work. We're also working on removal of microplastic fibers. These are the fibers that come from mainly washing machines, washing our clothes. These are kind of shedding fibers that again end up in the wastewater treatment plants, and we're working on that again. Dr. Roxana Suring is co-supervising that work, and we're working on determining the fate and removal of microplastic fibers. We've also worked with the Urban Water Research Center on a gap and a knowledge synthesis project with Dr. Kimberly Gilbright from Chemistry and Biology and Dr. Patricia Hania from Law. And we looked into the knowledge gap, whether in the scientific gray literature or indigenous knowledge on what do we know about emerging contaminant, what's been done about emerging contaminant, and what needs to be done in emerging contaminant in a knowledge synthesis project. We've been able to interview different stakeholders from the government, from municipalities, from operators in wastewater treatment plant, engineers, policymakers, so that we can understand more about this and what can be done from an engineering perspective, a science perspective, and also from a law perspective and water governance. And the next steps in our project is to focus on the science aspect, the engineering aspect, in determining, like I mentioned, what are these contaminants and what are the transformation byproducts that we're having, what are the removal efficiencies, and how can we conserve the system. And also we are trying to work on more collaborative and interdisciplinary and multidisciplinary projects so that we will be able to bring different people onto the table that speak science, engineering, social aspect, and legal aspect, so that we can tackle this problem on I would say, a more holistic approach, and let's stay away from the silos of only working alone to have this problem, so that we can mitigate the impact of this and also work on mitigation of that and the impact of these contaminants and how it's related to wastewater and climate change. I guess that's it. Please feel free to contact me if anyone is interested about these projects, and I'll be happy to share more information.
Hi, my name is Zanina, and I'm a PhD student at Toronto Metropolitan University. The title of my project is PFAS Removal in Biological Wastewater Treatment Plant. Now let me break this up for you. PFAS are man-made compounds that are used in a multitude of products, such as cosmetics, cleaning products, carpets, firefighting foams, and even takeout containers and Teflon pans. And you can imagine that all that waste from the manufacturing or from us simply cleaning our dishes at home goes into wastewater treatment plants. And that's why wastewater treatment plants are a crucial step into understanding the PFAS removal and kind of mitigating that pollution, because ultimately PFAS are toxic to human and environmental health, and we want to prevent them going into the environment and accumulating there. What I'm trying to do with my project is actually remove PFAS using biological treatment, which is rendering these chemicals into their simplest form of CO2 and water and eliminating any harmful byproducts. What you see here is I am trying to cultivate aerobic granular sludge and compare the removal with granular sludge to floccular sludge and see which one is better at absorbing or degrading the PFAS. And what's crucial with my project is that PFAS are a group of almost 5,000 identified compounds. So you can imagine that it's very hard to treat them and identify them. So I have found 4 target compounds that I will use in my project, and I will test the removal using aerobic granular sludge and aerobic floccular sludge. And ultimately, I want to try to mitigate the PFAS pollution inside wastewater treatment plants so that they don't go into the environment, and I am protecting the environment and human health with that.